Hello and welcome back. Hi Rajiv.
Yes, sir. How are you? Can you hear me?
Yes, good, good,
Okay.
good. So just we will.
For the presence of broadly neutralizing antibodies. Recently, her lab has successfully isolated a broadly neutralizing human monoclonal antibody from an HIV-1 clade C infected elite neutralizer that has been patented and published in the Journal of Virology. This antibody is currently being tested for its ability to neutralize the circulating clade C viruses in infected children and adults in India as a part of an ongoing Indo-South Africa project to evaluate its potential as an anti-HIV reagent. So I hand over the session to you, Dr. Luthra.
Thank you, Dr. Anand. I hope I'm audible.
Yes, you are, ma'am.
So we begin this postprandial session with the first talk by Dr. Rajiv Kumar, who's an assistant professor at the Institute of Medical Sciences, Banaras Hindu University at Varanasi. His research interests are to understand the immune mechanisms underlying the progression of diseases such as malaria and visceral leishmaniasis, and to distinguish anti-parasitic host immune responses that control disease from the responses that cause the disease. So that's a very important distinction that needs to be established. And he has a broad background in both cellular and molecular immunology of infectious diseases, as well as significant field and epidemiological work experience. He received training at the National Institute of Allergy and Infectious Diseases at the NIH in Bethesda, USA, and at a medical research institute in Brisbane, Australia. He has several publications in reputed journals such as Nature Immunology, Cell Reports, Nature Medicine, PLOS Pathogens, to list a few. And his lab is funded by many national and international grants. So today his talk is scheduled on the immunoregulatory networks in protozoan infection. And I request Dr. Rajiv Kumar to begin his talk. Thank you.
Am I audible?
Yes, please.
Um, just a minute. Can you see my slides?
No, you need to share your screen. I think it is about to. Yes, we can see now. You can come on the slideshow mode. Yes.
Okay. Thank you, Professor Kalpana, for your kind introduction. And I'm also thankful to the organizing committee of this immunology course who provided me an opportunity to talk today. So basically, for last 2 days, we have heard a lot about innate and adaptive immunity and T cell and differentiation. And even this morning, Dr. Avasthi and some of the speakers from Australia, they talked about the T cell differentiation. So what I'm going to talk is to show you how this information is needed and very critical during the protozoan infection. So as we all know that CD4 T cells response are critical for controlling any sort of infection. I'll be focused just on the protozoan infection. So what happens when there is any protozoan parasite entering the host? And these dendritic cells, they recognize the pathogen associated molecular pattern with the help of TLRs. They becomes activated. Although they are professional antigen cells because they constitutively express MSC and co-stimulatory molecule on their surface, but once they recognize those pumps, the expression level of MSC and co-stimulatory molecule goes up. So this disease, they start chopping those pathogens into smaller peptides, and those peptides then bind with the MSC class 2 molecules. They come onto the surface, and then this MSC peptide complex is recognized by TCR from the CD4 T cells. And depending upon this infection and the cytokine environment, these CD4 T cells then converted into the Th1 cells. And these Th1 cells then start secreting pro-inflammatory cytokines like interferon gamma and TNF alpha, which gives signals to the macrophages where most of these parasites, intracellular parasites, reside. And this macrophage starts secreting reactive oxygen species or reactive nitrogen species, which has the microbiocidal function, and then they kill the parasites. So although this response is needed for the control of infection, but this response need to be tightly regulated. If not, we will see immune mediated pathology as we see in the case of autoimmune diseases. So basically, the concept is there is a tight relation between host and pathogen, where pathogen requires an environment that allow its growth and development, while the host need an adequate immune response uh, to eradicate the pathogen without causing the self harm. And this requires, that means without causing the self harm, this requires the establishment of immune regulatory network, which ultimately determine the magnitude of immune response following the infection. And one of the key player in this immune regulatory network is the regulatory T cells or Treg cells. We have heard a lot about Treg cells this morning. So basically Treg cells are critical subsets of T cells that mediate the peripheral tolerance. It is of two types, we know. One is the natural Treg cells that is derived in the thymus and then comes into the periphery. But there is again induced Treg cells which are produced from the naive CD4 T cells in the peripheral after the exposure to antigen or the cytokine environment present over there. So this Treg cells, as Dr. Amit and others they talked this morning, this Treg cells was identified in 1995 by Sakaguchi and colleagues, and they told it as a CD4 positive, CD25 positive regulatory T cells. But the problem is that these Th1 cells, once they are activated, they also express CD25. So CD25 cannot be used as a marker to differentiate these Th1 activated Th1 cells from the regulatory T cells. So in 2003, Sakaguchi and Rudensky and their group, they identified a transcription factor called FOXP3. So FOXP3 is a master regulator of T regulatory cells. Now, we have heard this morning and even yesterday that this regulatory T cells show the immunosuppressive action. Now, how these Treg cells mediate this immunosuppressive function? So there are basically two mechanism, proposed mechanism. One is the contact dependent mechanism, other is the contact independent mechanism. So in contact dependent mechanism, what happens, this regulatory T cells, they express very high level of CTLA4, and this CTLA4 is an inhibitory molecules which has high affinity for B7 molecule expressed on the dendritic cells. So in normal condition, when DC interact with CD4 T cells, this TCR-MSC interaction as well as B7 CD28 interaction drives the activation of CD4 T cells. But when Treg come into the picture, it now occupy B7 molecules, so that there is lesser signals for CD4 T cells or Th1 cells for their activation. Apart from that, when there is persistence antigen exposure, Th1 cells also start expressing this CTLA4, and this CTLA4 then interact with B7 molecule expressed on dendritic cells, so that there is less activation. And not only CTLA4, once these Th1 cells are activated, they start expressing B7 molecule itself. So B7 molecule on CD4 T cells or Th1 cells then start interacting with CTLA4 present or Treg cells that suppress the T cell function. Again, in contact-dependent mechanism, when this Treg population cells, when interact with dendritic cells, it drives the expression of an enzyme called IDO, which is indole acetate 2,3 dioxygenase, and it catalyzes the conversion of an important amino acid tryptophan into kynurenine, and kynurenine then provides the immunosuppressive environment, and it also drives the expansion of regulatory T cells in the periphery. So by contact dependent mechanism, Treg cells show their immunosuppressive function. But in contact independent mechanism, these regulatory T cells, they start expressing immunosuppressive cytokine TGF and TGF beta and IL-10. So this TGF then binds with the TGF receptor present on the Th1 cells, and this TGF receptor interaction leads to the suppression of IL-2 gene. And you know IL-2 is required for the maintenance of those Th1 cells in the circulation. So in absence of autocrine IL-2 signaling, those CD4 T cells moves towards the apoptosis and they die. The other immunosuppressive cytokine, which is IL-10. So IL-10, it acts on the dendritic cells, and basically it downregulates MSC class 2 expression, co-stimulatory signals, as well as the interferon gamma receptor expression on the dendritic cells, thus preventing its maturation and activation, and hence the maturation and activation of CD4 T cells, because DC CD4 T cell interaction is critical for their activation. But what we have found in this protozoan infection, that these Treg cells are not the major player. Rather than interferon gamma producing Th1 cells, which secretes IL-10, is the major player to suppress the antiparasitic immune response. Why I'm telling this? Because many of the studies where people have removed those Treg cells, and they do not see any improved CD4 T cell activation. So just to clear it, what I'm telling is that when DC CD4 T cell interaction takes place, this DC secretes IL-12. That IL-12 drives the proliferation of CD4 T cells and their activation, and then this CD4 T cell start expressing interferon gamma, which is required for the microbiocidal effect of the macrophages through the induction of reactive oxygen or reactive nitrogen species. However, when there is persistence antigen exposure, this response need to be regulated. And in that case, what happens? This gamma producing CD4 T cells start expressing IL-10, which gives immunosuppressive signals to both CD4 T cells as well as the antigen presenting cells and the phagocytic cells. And this gamma producing IL-10 producing CD4 T cells are now designated as TR1 cells. And what we have found in case of the visceral leishmaniasis patient, that these TR1 cells are accumulated at the site of infection, and they are the major producer of IL-10. So what I talked here, we can see in here that when we collected the splenic aspirate cells from the visceral leishmaniasis patient, and when we cultured in presence of anti-IL-10 monoclonal antibody, we not only show the reduced parasite number, but enhanced Th1 cell response in terms of increased interferon gamma secretion as well as TNF alpha secretion in those culture supernatant, suggesting that IL-10 coming from TR1 cells are not only suppressing the Th1 response, but they are also promoting the persistence of parasite. Now, the question is, what is driving those IL-10 producing TR1 cells during those protozoan infection? And this morning, Dr. Amit talked about IL-27, and he has worked a lot in this field. So IL-27 is one of the important regulator of TR1 cell IL-10 production. IL-27 is basically composed of two subunits, that is P28 and EBI3, and it signals via its receptor, which is composed of IL-27 receptor alpha chain and GP130. So IL-27 receptor alpha chain is specific to IL-27, but this GP130 is shared by other cytokines, for example, IL-6. And what we have found that when there is persistent infection, the interferon gamma coming from CD4 T cells, they give a feedback signals to the dendritic cells and macrophages to produce this IL-27, and then this IL-27 drives the production of IL-21 by CD4 T cells, which acts in an autocrine manner for the production of IL-10 by Th1 cells. So what I talked here, you can see when we collected the blood samples from the visceral leishmaniasis patient, and when we stimulated those blood cells in presence with the leishmania antigen in presence of IL-21, we found increased IL-10 production. But when we added those exogenous IL-27 along with IL-21, there is further enhancement of antigen-specific IL-10 production from the whole blood of the visceral leishmaniasis patient, and this response was completely absent in the endemic control, suggesting that this IL-27 and IL-21 promote antigen-specific IL-10 production. Now, apart from IL-27 and IL-21, there are various transcription factors which drives the expansion of IL-10 producing TR1 cells, and I tell it as transcriptional repressor. And some of the important transcriptional factors are aryl hydrocarbon receptors, BLIMP1, which is B cell induced maturation protein 1, CMAP, and EGR2. So IL-27, IL-21, along with this transcriptional factor, that drives the expansion of Th1 interferon gamma producing Th1 cells into interferon gamma IL-10 producing TR1 cells. So just to summarize, what we have seen is that in presence of persistence in parasite antigen exposure, IL-27 from dendritic cells or macrophage stimulate state 3 and state 1 independent transcription of CMAF and aryl hydrocarbons in Th1 cells, and then this CMAF and AHR combines together and they bind to the promoter region of IL-10 as well as IL-21 to drive their transcription. Apart from this IL-27 along with TCR signaling promote the expression of transcription factor EGR2, which is critical for the induction of BLIMP1 and generation of IL-10 producing Th1 cells in a STAT3 dependent manner. So two things are happening here. This IL-27, which is coming from DCs in response to the interferon gamma, are driving STAT1 and STAT3. This STAT1 and STAT3 then regulates the transcription of CMAP and AHR. And this combines together, binds with the promoter region of IL-10 and IL-21 and drives their expression. This IL-21 then acts as an autocrine manner. It acts as an autocrine growth factor for IL-10 producing Th1 cells. So this comes from these Th1 cells, act on these Th1 cells and drives the production of IL-10. And then this IL-10, which is produced by these TR1 cells, can suppress the inflammatory function of Th1 cells and phagocytes, but also suppress the antigen processing capacity of dendritic cells and the macrophages. So there is a complex network which operates during those parasitic infections, where a lot of cytokines, lot of feedback mechanism, as well as lot of transcription factors are involved to drive the expansion of IL-10 producing TR1 cells. In recent time, apart from IL-27, type 1 interferon has emerged as an important player of CD4 T cell regulation during the parasitic infection. We all know that type 1 interferon are very critical during the viral defense, but what we have found in our control human malaria challenge study that we conducted in collaboration with, during my postdoc, I worked with Dr. P. Singh-Varda in QMR, Australia, and I'm still part of this program. So what we did that we recruited the healthy volunteers and we infected them with a low dose of Plasmodium falciparum, and then we tried to study the generation of immune response. And what we found that during early phase of infection, type 1 interferon emerged and it suppressed the innate immunity. So basically it suppressed IL-6 production and IL-17 expression by the monocytes. But later during the infection, day 14 post-infection, what we found that this type 1 interferon not only suppressed the Th1 response by reducing interferon gamma and TNF production, but they also drive the expansion of these Th1 cells into IL-10 producing TR1 cells, and then this IL-10 suppress the Th1 function. So what I said here is, you can see here, this is just a getting strategy to show how we look for the TR1 cells. So basically this population, which is IL-10 positive, interferon gamma positive, is the TR1 cells. And what we found that when we collected the PBMC from those healthy individuals infected with Plasmodium falciparum, and then 7 days after drug treatment, when we took the PBMC and we stimulated with parasitized RBC, we found increased number of TR1 cells, which was reduced when we blocked this type 1 interferon signaling using anti-IFNAR alpha monoclonal antibody. And once we blocked this type 1 interferon signaling, we found that there is reduced IL-10 production. And once we blocked this IL-10, we found that the interferon gamma and TNF alpha level goes up, suggesting that IL-10 coming from this TR1 cells is suppressing the interferon gamma and TNF production by Th1 cells. Similar to this study, recently we conducted another studies in a different disease, that is visceral leishmaniasis. So we collected CD4 T cells from VL patients and endemic controls. We isolated PBMC, then enriched the CD4 T cells using the magnetic base and went for the RNA sequencing. And what we found that type 1 interferon signaling emerged as a most potent negative regulator of CD4 T cells in those VL patients. This study we published in Cell Reports a few months back. So just to clear what we found, that the good part of this study was that we showed that how we can manipulate this type 1 interferon pathway to treat or to enhance the antiparasitic immune response. So what we did, we used a established animal model of the infection, and we infected mice with the L. donovani MST goat, and then on day 14 we treated those mice either with the conventional drug, optimum or suboptimum dose of conventional drug, or in conjugation with the type 1 interferon receptor blocking antibodies. And you can see that once we combine the low dose of drug with the type 1 interferon receptor monoclonal antibody, we see a reduced parasite number both in the liver as well as in the spleen. Most importantly, we identified a small molecule inhibitor called ruxolitinib. So ruxolitinib is basically inhibitor of JAK1 and JAK2. And once we use this small molecule inhibitor instead of type 1 interferon receptor monoclonal antibody, we see more potent effect. We saw, like you can see here, a reduced parasite number in the liver as well as in the spleen. And once we combine this small molecule inhibitor with low doses of conventional drug, that is Ambisome. Ambisome is lipid formulation of amphotericin B. We saw that there is synergistic or additive effect, because here we can see more pronounced reduction in the parasite burden both in the liver and in the spleen. And given with the fact that the host directed therapy aimed at improving the control of infectious disease are likely to be used in association with the conventional drug, these informations are very critical, because we used this we reduced the amount of drug, but we added a small molecule inhibitor and we found a better parasite control. So from here we moved to the human side and we collected the blood from the VL patient, and we performed the similar study. So just before the start of treatment, we stimulated the whole blood of the VL patient with this Leishmania antigen in presence or absence of the ruxolitinib. But you can see we could not find here any difference in terms of the interferon gamma production. And we thought that maybe once drugs will be in the circulation, the more parasite will be released and more type 1 interferon signaling will be activated. What we did that 5 days post infection,